Today, I'm going to talk about how we can go from multiple sequence alignments to phylogenetic trees and arrangements of evolutionary or depiction of evolutionary relationships between sequences and organisms from which those sequences are derived. And in this case, one very interesting example that I'm going to talk about is the first warning that a pandemic is going to come into the world, which is the outbreak of the SARS virus. This is currently the COVID-19 is actually called SARS-CoV-2. It's the second version of this virus, but the first version of this virus hit the world in around 2003. So these are the outline. I'm going to talk about how this outbreak happened and what lessons we can learn from it. And then I'm going to basically, this will be the motivation. And then from there, I'll be telling you how we can organize multiple sequence alignments into evolutionary trees through distance matrices, how we can come up with algorithms to construct these trees. And there I'll be mostly talking about focusing mostly on three to four algorithms. Three algorithms I'll go in detail: additive phylogeny, ultrametric evolution trees, and maximum parsimony. And then I'm going to end with what are the modern tools that are used to reconstruct phylogeny. All right, so this hotel looks quite straightforward, like a very standard hotel. This was in Hong Kong. So this is the Metropole Hotel, and on I think the 13th floor of this hotel, a doctor, a Chinese doctor stayed, and he came back from treating some patients in a village, and he was attending a wedding, and within a few days he fell very sick after coming back from the village, and he checked himself into a hospital close by from this hotel, and very shortly he died. So he was the first documented case of the SARS-CoV outbreak, or a viral outbreak. Soon, a lot of people who were staying on the same floor, all of them got infected. This hotel was finally closed down, and actually, this hotel even features in a very popular Hollywood movie called Contagion. I highly recommend you to see it to get motivated about this problem. So this virus, SARS virus, the first one was, it took a few days to go from this hotel to all
So
around the world. Yeah.
this first virus was also named SARS-CoV.
Yes. It at this point of time, it was not called SARS, but they started to call it SARS because there was full form is severe acute respiratory syndrome, right? So after the outbreak, after they had found what was happening, then that's when this is the first time the virus was called SARS. So this is the first version of it. So if we go back and compare previous outbreaks, so one of the largest previous outbreaks this is documented in history. I think most of us even hear it in NCRT or ICSC textbooks in school, is the Black Plague. So Black Plague, if you can see it, started in around in I mean, it started around in Rome. They say that it came from Asia. And then slowly and slowly, you see green areas, it spread to the green areas. It took 1 year. Then it spread to further north of Europe and Africa in um, in 2 years, right? And it further spread to Russia and specifically this particular region where you have a lot of activity happening currently. And it took about 4 to 5 years for this outbreak to happen, right? But nowadays, if you actually look at it, as I don't know if you're following the news, a lot of students are even getting evacuated by this route. You can go from Kiev all the way to Istanbul, right? Romania in a few hours, right? So therefore, nowadays these outbreaks are happening much fast, and I think there is no. Um, I don't need to more tell you everybody how these things, how quickly they happen. If in one colony or in one building you will have one COVID-19 case, most likely you'll see that the entire building is going to be infected within a week's time. So this is what happened in the case of SARS-CoV. There were first cases were found in this Metropole hotel, and within a few weeks' time, this already crossed over to the Americas. It crossed over to the US and Canada, and very soon it was spread across all over the world. Right. So this essentially was, and it was thing for a lot of movies. So this was basically caused by a virus, which we call the coronavirus, because it has this kind of corona-like shape, similar to what happens when you have a solar eclipse, uh, and the sun basically forms this corona. So this is a part of the sequence of the coronavirus, and because this is an RNA virus, so while copying of RNA, the machinery that does it, the RNA-dependent RNA polymerase, or when this RNA gets copied into DNA and there is a reverse transcription happening from DNA to make more RNA, this process is very faulty, unlike, let us say, copying of DNA into DNA, right? Because DNA polymerase has a polymerase activity, and this is having a proofreading activity. So in most RNA polymerases, there is proofreading activity is typically missing. So what happens as a result of that is, what a sequence originally that looks like this gets quickly starts to accumulate mutations, as you can see in red, and these mutations typically favor the virus to spread more, and to, you know, cause sort of get normalized in infection. What sort of what we are finding in COVID-19, that initially the virus was very virulent, and now slowly and slowly the strains are getting assimilated into the population, because for virus it's more important to survive. The way that the virus survives is to actually stay in the host and not kill the host. But typically, what you see is that the virus keeps on accumulating mutations, right? And you would essentially see many different versions of the virus uh, come up as the outbreak spreads. So initially, people were trying to understand exactly where did the virus come from. And before the SARS virus, there was a big virus, another virus that had an outbreak. That was the bird flu, or H1N1. And people were trying to figure out, where the virus came from. And this is going to be essentially what we are also going to talk about. So the questions that we are going to try to answer in this module is, which animal actually gave us SARS? Who were the people who were first infected? Like, how was the first human infected? And how did actually SARS spread around the world? All right. So all these questions relate to the idea of constructing evolutionary trees, which essentially go back to look at the original sequence of the virus, and then seeing how the different mutations are getting accumulated over the course of the virus replicating itself, and how these downstream mutated viruses, how can you trace back to the original virus, right? So when you talk about spread of virus, SARS virus around the world, the way that you can say that, okay, India was the origin of, let us say, the Delta variant, was because the sequence of the Delta variant was first found in India, and then later on it was found in other places. Right. So who was the first infected? The first person that was infected seems to be the people in Wuhan, right? In China, for example, I'm talking about COVID-19. So if you turn back time, not only the first cases came up there, but the virus that was isolated at that point of time, or viruses from those patients, they had uh, they had much less kinds of mutations. They were, let's say, the original viruses, right? That were infecting the people at that point of time. And now, as the virus spread, it traveled from different parts of the world. That led to the virus mutating, right? And that's how we are actually tracing the virus itself. And which animal gave us the virus? If we turn back the clock, people have tried to look at try to sample viruses from bats. They try to sample viruses from different other animals which were eaten in China, as I mean, it's allowed to eat wildlife there, in order to meet the like the food shortage. So people try to isolate viruses from the different animals that were there in the Wuhan market, and that's how they were trying to compare the sequence of the virus from that animal and the virus that was isolated from humans, and they try to see, okay, are there these sequences? Do they look very similar when we do sequence alignment, as I have shown you in the earlier module? All right. So the same things, all these ideas were actually already prototyped to, or all these modules were set up already when the first outbreak of SARS happened. And the basis for answering all these questions again is our ability to make evolutionary trees, also called as phylogenies, from the virus sequence. So this is one of the first phylogenetic trees that was drawn by Darwin. And if you have looked at the video that I put in the talk in the last module about multiple sequence alignments, you would realize that these are three different kinds of finches, which are birds which were isolated in the Galapagos Islands, where the birds, because of geographical isolation, these are islands which don't have any other, which are surrounded by water on all the sides. That's why it's an island and quite far away from mainland, because of which birds started to evolve to have different feeding habits, because of which they had different evolutionary phylogenies. But the idea of this kinds of trees is that all these birds actually are rooted and they came from a common ancestor that was found on regions, the mainland, which was away from which was close to the island, right? So the whole idea of phylogenies is that every organism, be it closely related organisms or organisms which are further apart, they are related to each other because they share common ancestors, and they are sharing common DNA or genetic material from which is getting inherited from one generation to the other generation until the two new species are happening. And while they are getting inherited from one generation to another generation, because of the mutational processes, which are, some of them are random, some of them are acquired, these mutation processes actually lead to these sequences look more further and further different. So if you would compare the sequences in closely related species, they will be similar. Sequences which in distant related species, let us say D and C, these sequences will be different. All right. So let me just quickly ask how many people did actually go and see that video? I mean, it was quite a short video from multiple sequence alignment. Just for me to understand whether putting such videos is important. A show of hands, please. Okay. All right. So yeah, I would hope you do see it so that there is a continuity in what I'm teaching so that you can understand what is being taught. All right. So this is an example of trying to answer the first question, which animal gave us SARS. So in order to do that, let's take a look at another, so not an outbreak, but another viral infection, which has jumped from humans, jumped into humans from other species. So this is the HIV. So this is human immunodeficiency virus. So as you can see, uh, if you take the sequence of HIV strains isolated from lots of primates, so humans, chimpanzees, and different kinds of monkeys. So this is a simian immunoacquired immunodeficiency virus. So the HIV in monkeys is called SIV. Simian is monkey essentially. You see that in a lot of places you have the virus actually having common ancestors, or having commonality with a particular being very similar to a monkey. So this is Sooty Mangabey SIV, and that's therefore people say that okay, most likely there was some virus, common ancestor virus that was infecting all these different kinds of monkeys, and from these monkeys actually they jumped over to humans. So HIV A and B are the most prevalent kind of strains of HIV which are found in the world. And it was later found out that there were more some practices where people, some tribes in Africa, they were actually eating the meat of these of monkeys and so on and so forth. So now Basically, the idea, given that I've given you some motivation about how these kinds of what kind of questions we are asking and how these phylogenies would help you answer these questions. Let's go into transforming a multiple sequence alignment
Sir,
into what's called a distance matrix. Yes.
sir, they were eating monkeys, so they got HIV. So, sir, SIV and HIV are different. So then, how can they go get uh, like
So,
SIV?
so SIV and HIV are the same virus. In monkeys, it is called simian immunodeficiency virus. The same virus in humans is called human immunodeficiency virus.
So name is different. Sequences are same.
Sequences are very similar, yes.
Okay, sir.
So similarly, when you talk about COVID-19, right? If I will discuss, so those viruses are typical, right? Those viruses would be called based on the animal from there they are isolated. So they would be, it will, you will call it a bat coronavirus. Okay. If it is isolated from a bat, but the virus looks very similar to the human coronavirus. I'll come to that. All right. But here people did not know, right, that these two viruses are related until they actually saw, until they actually made these kinds of trees and made multiple sequence alignments. Okay. Is it clear? Yeah, Bhavya.
Sir, I have a general question. Like, do people sequence the like for COVID? Do are the sequence of every patient is sequenced?
No. So because sequencing, the act of sequencing is quite an expensive process. I mean, not very expensive, but expensive enough. It takes about 10,000 rupees to sequence one COVID sequence, right? So you can imagine if there are like crores of people getting COVID, specifically in India context, therefore India lags behind in the sequencing. Uh, the number of sequences generated per for the number of people we have, but it's a randomly people select. Our people are selected based on their symptoms, based on where they are coming from. So sometimes if you're coming from, if you'll see on Delhi airport, if you take an international flight, you will be randomly sampled, and there some of the samples in Delhi at least, they go to National Institute of uh, National Institute of Tropical Diseases, and another set of sequences go to IGIB, which is Indian Genomics Integrated Institute. Yeah, Ratnesh.
Sir, this is also like related to COVID. So, sir, in once in a while, like I was seeing in the news that here at KSBS, in January 2020, like Professor James Gomes and etc. They have done this coronavirus sequence with something like sequence alignment, and they published some research paper on that
It was
this
retracted. Yes. Yeah.
yeah that this virus has similar sequence to some other, so
Yes.
I don't know about more about that. Yeah,
I'm
so sir,
waiting for your
that
question.
was done multiple sequence alignment, right?
Yeah.
Okay, sir. Okay.
So what is the question?
So sir, I was just wanted to know that was it multiple sequence alignment or something else. So,
So now next class you will tell me what it was.
okay, sir, then I will
Yeah.
see.
Read the paper and tell me. All right, Bhavya, is the question solved, or can you keep your hand down?
Yes,
Or
sir. I have a follow-up question. Like,
yeah.
how did the Chinese know to sequence the COVID-19 patient? Like, they also randomly, like, they got some new symptoms and they started sequencing it, or there
So
is.
this is now because I mean China has been the center for Hong Kong area, has been center for a lot of outbreaks, right? So that causes a lot of damage, right? Economic damage, and the flights stop and your economy stops. So they had the bird flu, they had SARS virus also. So, and when specifically when patients are not responding to medicine, right? So let us say tomorrow in our in IIT, right? You get some virus, right? People are falling sick. If the doctor gives them medicine and the person is cured, then it's fine, right? But if the person is dying very quickly, if some, I don't know, God forbid something like that happens, but if if you are not able to control the symptoms of the patients with known drugs, then nowadays, therefore, nowadays, like sequencing is one of the prime go-to methods to understand exactly what is the person infected by. And these are the kind of problems that I think a lot of people are solving around the world now, is how can we actually identify the exact species which is infecting you or causing you the problem. It's not only for viral infections, but also for bacterial infections. Make sense? Shall I move ahead?
Yes, sir.
All right. So this is how an alignment looks like. So in this case, I'm showing you a ungapped alignment, but you can imagine that this can very well represent a gapped alignment. And then whenever you have, you count the columns that are common between two aligned sequences, and you can come up with a distance matrix like this, right? So basically the number Dij here represents the number of differing symbols between the organisms in ith row and jth row. So for example, chimp and humans, the distance here is 3. That means there are three locations, three columns where chimps and humans are have differences. So you can probably see the second column here, um, and second last column here. There should be one more column that should be different. Yeah, and this column here, right? So this basically is a distance matrix captures what is the similarity. So the more sequences are similar, the value will be more closer to 1. Now we want to go basically from these kinds of distance matrices. So yeah, these are the differences are highlighted. So um, I think this is quite straightforward. How would you form a distance matrix? Can you come up with a algorithm for this? Can I see a show of hands if you have any everybody can come up with an algorithm for this with the tools that you have been given. All right, good. So this is how the spike proteins looks like. All right. And we are going to basically come up with the ways of looking at the spike protein. So spike protein essentially is the protein in the coronavirus. So in the corona of the virus, in the outer membrane, which goes and binds to the human cells using another protein, which is called the ACE2 protein. So, we basically want to go from this kind of a distance matrix to such trees. So what are trees? Trees are basically directed acyclic graphs. So it's a graph because you have nodes and you have edges connecting these nodes, right? There are no cycles here, so therefore it is acyclic. It is directed because it is basically going from top to bottom. So this is the tree of life here, as you can see. So here it's a snapshot of tree of life. So life started from a last common ancestor. So this is called Luca, uh, last common uh, last common ancestor. Yeah,
Last
LC,
universal common ancestor.
last universal common ancestor. And then you had bacteria, you have archaebacteria, you have protists. Sorry, you have eukaryotes coming up. Then from eukaryotes you had protists. Then from there you had plants, right? So all of them are sort of organized in this kind of relationship. The other property that you see, that the organisms that you are currently observing in present day, these organisms are at the ends of this tree, right? And they are called leaves, and they are represented by having degree equal to 1 because they are only connected by the last common ancestor between two species. All right. The other thing that you see is all the internal nodes, which are ancestral species. They typically have a degree greater than 1. When they are just last common ancestors from present day species, their node value is 3. And if they are the original ancestor, that is the root node, that is the node which is at the top, they have, you see the degree is typically 2. All right. So this is
Sir, can
uh,
you please explain again what is degree here?
So degree is the number of edges which are connected to a node. So that doesn't change. So I think the concept of degree is common for all the graphs, whether it was a De Broglie graph or Hamiltonian circuit-based graphs, overlap graphs. Degree is always the same. The number of edges that are coming into or going out of a node. All right. So um, there are these two statements that we have to sort of think about. The first statement is that every tree with at least two nodes has at least two leaves. Is this obvious? If you have a tree with two nodes, it will have at least two leaves.
Yes, sir.
Yeah. So there are these two theorems about trees. One is that a tree which has at least two nodes is going to have at least two leaves, and a tree with n nodes has exactly n minus one edges. Can somebody tell me why? This one? So these are axioms. So that these are universal truths.
Like because n minus one edge, because there is only not a cycle in a tree.
Exactly. So each node is connected to only one other node, right? Sorry, each node is connected only once to the other node and does not have a cycle, so it cannot be n minus like more than n minus one edges. Sorry, my computer is slow today. All right. So now if we take a look at trees, these trees can be rooted. So the tree that I showed you, these trees, these are unrooted trees. You don't know which is the oldest node here, but rooted trees basically are trees in which the oldest node is at is one end of the page. It can be at the top of the page, or sometimes it is on the left side of the page. And so this is the most recent ancestor. So this is the oldest ancestor. And as the time goes by, you travel down the tree. So at the bottom of the tree where you have the leaves, these are all the species or all the sequences of these species which are observed in uh, the current day. So this is what's called a rooted tree. If you basically remove this node or you don't know, you basically flatten out this tree, then those trees are called unrooted trees. All right. So the algorithmic problem that we want to essentially solve here is that we would need to construct an evolutionary tree from a distance matrix. So the input is a distance matrix, which you may be obtaining from a multiple sequence alignment, and the output is an unrooted tree fitting this distance matrix. Now, can you define fitting here? What do you mean by fitting?
So least error, something of
That is when you're doing regression.
yes, sir.
Yeah. What do you
Sir,
mean by fitting here?
we can define fitting like what is the percentage of alignment, or if the score is high, we can say that it is more fit than.
So you are
Uh,
given a distance matrix, right?
yes.
You are given a distance matrix and that distance matrix is representing a tree. So when will a tree fit a distance matrix?
When the distance is low, very low.
Okay, so basically let us see that we have a distance matrix like this. When I say that you want to have a tree which fits this matrix, that essentially means the edges that you have between the nodes, they sum up to represent the distances that are represented in the matrix. All right. So for example, there were four species here. Let us take the species of whale and seal, right? The distance is 2. So now if you want to go from seal to whale, when you travel this graph and you'd have this edge is having weight 2, this edge is having weight 0. So the distance between seal and whale is 2. Similarly, if you want to go from human to seal, the distance is 7. So how will you calculate the distance? This is 2 plus 3 plus 2. All right. So fitting a matrix essentially means,
So I had one doubt here. Uh,
yeah,
so the shouldn't the whale spot coincide with the right gray spot since there's like zero distance between them?
sure, it should be. It should be. But we assume that seal and whale are having a common ancestor, right? This is just an
Yep.
example
So
matrix, but because they
okay,
are
okay.
not coming, right? This is the internal nodes are always nodes that are ancestral in nature. the not
So,
present day
but if
species.
like there was some distance matrix where like the distance was actually zero, so do you actually need to draw another branch from that node?
No, you don't need to draw a branch from that node, but I will come to the kind of
But
trees
in
that we are
that case,
working.
if the distance will be zero, can't we say that this is the same as species? Like if the sequence are same, then only the distance will be zero.
Yes,
Exactly.
but
So
you
like
don't know
we can
the,
say,
but you
so
don't know this species, right? This is an ancestral species.
but here can't we say that this implies that seals have derived, like have originated from whales?
You can say that if you compare it with respect to chimps and humans in this context, you can say that seals would have are closest related to whales. But whether you have the whale node here or whether you have the whale node here, that doesn't change, because the reason why you can make that statement is based on the distance between seal and whale. I mean, in this case, we are talking about seal and whale, right? Maybe there is one more species that we are not talking about right now, right? I mean, this is just a snapshot of the matrix,
Yes,
right?
sir.
You can have many species. And
Yes.
the other thing is that, so when we talk about this sequence of whale and seal, we know that whale and seal are different species. That's the case with most similar most trees like this. And they must have had a common ancestor from which both of them have derived. So you can not make a statement that some current day species, right, is an ancestral species to basically another current day species. So
So but
these
like
state.
here the ancestral
So
species
let me
and the
give you an
whales
example.
have,
Let me give you an example. So yeah, people basically say that dogs have evolved from wolves, right? That's
yes, sir.
probably a statement that you would have heard. But
Yes.
you cannot make a statement that a dog currently found has evolved from a wolf who is currently found. Okay. They have evolved, dogs have evolved from an ancestral species of wolves thousands of years ago. How did that ancestral species look like? We don't know. But most likely, it was more related to wolves than to dogs. The same thing you can say here.
So, so that is fine, but then in that case you'll have some sort of a distance between the like common ancestor and the like current species, right? But here, if you do not have any distance, like since this is a hypothetical scenario, but if you have like a zero distance between like the common ancestor and the whale, so can't you just say like the common ancestor like diverged into a seal and it uh, like basically then the common ancestor was a whale and a part or some say some organisms of those
Yes,
whales diverged into
I
seals.
agree. No, no, this I'm conceding the given the tree, this current tree, you could say that, but typically internal nodes are not observed in nature.
Fine, sir.
Yeah.
Like so basically we won't have this zero distance
You
like
would know,
most of the
you
time.
would rarely have the zero distance.
So
Fine.
in nature, we will naturally found DNA. Will we have like distances 1, 2? It should be like terms of thousands
Yes.
and 100,000. So
Do you
so
want me to solve? I mean, you must have prepared for IIT, no?
yes, sir. Yes.
You must have HC Verma sometimes, I don't know, most likely nowadays people still do HC Verma, right?
Yes, sir.
Does
Yes.
do they actually give you 9.8 G, or they give you the real value of G, which is 9.82 something something something?
Yeah,
Um,
sir.
I'm just
It
giving you an
depends.
example, right?
Yes, sir. Yes.
Give
Yeah.
10.
So basically, the idea of fitting essentially means that the tree and the edges represent the distances between the nodes, all right, which are given in this matrix, as I show you here. So the problem that we have is we have to compute. So there is two problems here. So one problem is how do you check whether the tree that you have been given is a fitting tree, right? So the distance between leaves problem essentially is you have to compute the distance between the leaves in a weighted tree. The input is a weighted tree with n leaves, and output is a n cross n matrix where d basically dij, where dij is the length of the path between leaves I and j. You essentially, if you solve this problem, then and you get a matrix, right? If this matrix is equivalent to the distance matrix that you have originally from which you are making the tree, then you have solved the problem. The original problem was given a matrix, you fit a tree. All right. So fitting essentially means that you obtain a tree with n leaves such that when you make an n cross n matrix where dij in the matrix represents the length of the path between leaves I and j in the tree that you have made, and this matrix is equivalent to the input matrix, then you would have solved the problem. But now you can all probably appreciate I've given you a circular argument, but we can now define what do you mean by fitting of a tree to a fitting of a matrix to a tree. Is this clear to everybody?
Yes, sir.
Yes, sir.
All right. And the other problem was construct an evolutionary tree from a distance matrix. So a distance matrix is given and you have to basically output a unrooted tree. All right. So, and quick exercise break. Can you quickly come up with a way to make a tree for this?
Sir, I think making a tree is very, very hard, and that for that we will have to only use like computers, and we can use make this
Can you
easily.
come up with some ideas here?
Okay, sir.
So can it be like starting from the shortest distance and then adding the ascending order?
Yes, that's one idea. What other ideas? So let us say shortest distance. So that's what kind of algorithm would that be out of all the algorithms that we have talked
Like
about?
from all the distance matrix, we will
No, but what class of algorithm would that be? That would be a
greedy.
greedy algorithm, right? Okay. So greedy algorithm is one algorithm. What are the other kinds of algorithm can we have? Can we solve this problem randomly? We take any two nodes, try to connect them and see whether it works, right? So you can solve this problem using some random algorithms as well. But the issue here is that if you connect two nodes, then it affects the distances between all the other nodes, right? If I, J,
Yes, sir.
K, L are four nodes, if you let us say connect K and L because of the shortest distance, that's going to affect all the other nodes as well. So basically a distance matrix, we have to first define whether you can actually fit a tree to the matrix. And the matrices for which those distance matrices for which a tree unrooted tree exists, which is fitting it, these kinds of matrices are called additive matrices. All right. So these are the kind of matrices that we are actually working with. So in this particular case, no tree will be fitting this matrix if you give it a try, because this is not an additive matrix. All right. Further, I showed you one example of a tree here. Can you make another tree here? Another kind of tree that fits this very quickly. So do you think this is also a tree which solves this problem?
So
No,
distance cannot be like 1.5, 0.5.
What about this? 1, 1,
Yes, sir. We can
right?
form one.
So this basically, if you start to solve problem like this, then you are going to end up in a basically you can have many trees and this problem is going to be intractable. So now we have to define another form of a matrix or another form of a tree. So the tree should be unrooted,
So
right?
isn't there some rule like there can not be a joining?
Exactly, exactly. So basically the kind of trees that we are looking for are the trees on the top. And these trees typically do not have nodes which are having degree equal to 2, right? So we are looking for trees which are basically having no node is having degree equal to 2. Typically we are looking for trees which have node degrees equal to 1, that is the leaves, or equal to 3 if it's an unrooted tree, or nodes which have uh, the ancestral node only has a degree equal to 2 in the case of a rooted tree. right? Is this clear? So this is the first kind of trees is what we are looking for, not the second kind of trees. Now, if you, Vishantan, to go to your question, if we were to actually allow the whale to be found here, then this would become a tree essentially, which has a node which is having degree equal to 2, right?
Yes, sir.
So this is not this is not allowed, let us say, because it makes our life very complicated. So typically you are looking for these kinds of trees. All right. So these kinds of trees are called simple trees. These trees which have no nodes of degree 2. And there is a theorem which says that a unique simple tree will fit an additive matrix. Okay. So this is a theorem. All right. So we can
Sir,
yeah.
can you please explain what is unique simple tree and additive matrix here?
So additive matrix is a matrix for which a tree exists. A tree can be fitted. A unique simple tree is a tree which has no nodes of degree 2.
Okay, sir. Understood. Understood.
So now we reformulate our problem to sort of be very specific. Distance based phylogeny problem. Basically, you have to construct an evolutionary tree from a distance matrix. The input is a distance matrix and the output is a simple tree fitting this distance matrix if the matrix is additive. All right. So now imagine when you're writing your pseudocode, you are essentially adding a few lines where you are checking whether the given matrix is additive and you're checking whether the output tree uh, is actually a simple tree or not. Right. So now these were the ideas. You can essentially transform your multiple sequence alignments into distance matrices, and then you can convert them into evolutionary trees. So trees which fit these distance matrices. Now let's try to go for an algorithm of converting these distance-based matrices into phylogenies. So one idea that Bhavya sort of suggested, let's go with the those two nodes which have the least distance. For example, here, right? You have seal and whale at the lowest distance. You connect them, right? And you go from there, right? So seal and whale are neighbors, meaning they share the same parent. So you say that, okay, they have the same parent. Now the theorem is every simple tree, at least with at least two nodes, has at least one pair of neighboring leaves, right? So this is this, I'm just restating the same idea. So now you have this. Now, can you, given that they are neighbors and their total distance is 2, is there some rational way in which you can compute these distances? Whether this is 2 and 0 or this is 1 and 1. Can you think about this?
We have to know about this their ancestor. If we don't know, we can have just like 0 to 1 1 and 2 0, nothing more.
Any other ideas?
So we will take a third organism and see from there.
Right? So we can essentially take a third organism, and based on the third organism, you can actually now construct linear equations, right? So let us say that you have, the whale and seal, you assume that they are neighbors because they are close to each other. This is an assumption, by the way, let me tell you. All right. You say that they are neighbors, that means they would have a common ancestral node, right? And now you take a third species, you know the distance of this third species from whale and seal, right? These two other species. So you have these three distances, right? So you have three distances and you have 1, 2, and 3 and 4 unknowns, right? So let us say you have only one, you make it 1, 2, and 3 unknowns. So distance between the internal node and the third species becomes a 3 unknown. So you will have 3 equations and 3 unknowns, right? So these linear equations you can solve, right?
Yes, sir.
Makes sense to everybody? So essentially, when you solve these 3 linear equations, you can essentially get these distances. So how does it work? Let's see. So let's say seal and whale are neighbors, and let us take a third organism. Let us say, so they are neighbors, so they are having an internal node M, And let us basically denote these things now. So distance between M and seal is seal comma M. Distance between seal and whale, sorry, M and whale is given like this, right? And now these are the first set of equations that you have. So the distance between seal and M can be represented like this. Now let's take a third species. A third species, let us say it is chimp, right? Now, based on the value of distance between chimp and seal, you will get this kind of equation and you can plug in the values and now you'll get the value here of seal comma M, right? Because this is 2, automatically you know this value will be
0.
0, right? Because you know the distance between seal and whale, and this, so this sum of this and this should be equal to these two distances. So you solve. So basically by solving this, you now start to get the values, right? So this is the first node that you added like this. Now the next thing that you can do is you go back and you say, now if you see that human and chimp seem to be having a value which is lower than 4, which basically means that they must be neighbors, not uh, not M and chimp, right? So now what we do is you can add, right? You can add, basically you can calculate the things like this. Uh, Sorry, I think I missed a step. So once you have added this value for the chimp, now you can add the value for human as well. So the distance between human and chimp is 3, but distance between human and seal is 7, human and whale is 5. So this is 2 here, this is 0 here. So if you add an edge of length 5, you are essentially going to complete this matrix and you will have one internal node, which is M here, right? But now we know that the distance between the human and chimp is 3. So that basically means that human and chimp should be neighbors. So therefore you put another internal node, right? So you just remove this part of the graph, you put another internal node, let us say A, you get those same equations, you solve those equations, and now you have solved the distance between human and chimp. And in order to find the distance between A and M, you already know all the other distances. So you know that between chimp and seal, there was a known distance, and based on those known distance, you can actually find the length of the edge A and M as well. Right? So quickly, can you implement the same idea to this matrix quickly? Apply this recursive approach to the matrix given. Anybody giving a try? I really want you to give it a try because there is something that I want you to tell. You will find something which is weird when you apply the algorithm.
Sir, is it not additive?
It is additive. Anybody? You will not get more than this time in exam to solve this. Anybody got negative edges?
Sir, I am in one equation. I am getting that 0 is equals to 9.
Okay. So I think that's probably not going to be possible, but what you'll see is that in one of the cases, you are actually going to get a negative edges. But to answer um, Bhavya, your question was that this is an additive matrix and there is a tree which fits it, as you can see with this tree. You can check,
Sir, there will be a negative matrix. Something.
right? So
Yes, sir.
can all of you check? Maybe does this tree fit?
Yes, sir.
Right? So this is an additive matrix because a simple tree is fitting this matrix. If we use the assumptions that we were going for earlier, which was an obvious, the greedy strategy as Bhavya suggested, we will go for the first try to assume, take the minimum element, right? And we will assume that the minimum element is actually neighbors, right? But do you see that are they neighbors?
No, sir.
No, sir.
Right? J and K are actually not neighbors. They are having two common ancestors, in fact, right? Ancestral nodes. So this assumption that we had for making the tree is not accurate, right? So instead of actually looking and trying to fit neighbors, we should now modify our approach and we try to compute the lengths of the limbs that would be found between two nodes. All right. And the idea there is that you still have these same equations, right? But now instead of trying to solve them based on the assumption that the nodes which are closest to each other are going to have ancestral nodes, we are going to take all possible combinations of ancestral nodes. All right, what do I mean by that? So here we are assuming that I and J are neighbors, right? You assume that I and K are neighbors. You assume that I and, let us say this is L, I and L are neighbors, and you compare all the possible distances between I and K, given that I and J are neighbors, I and L are neighbors, and I and K are neighbors. All right. So the limb length theorem essentially says that the length of the limb I is equal to the minimum value of d I k plus d of I j minus d of j k for all the leaves k and j.
Sir, can't it be like three of them are neighbors and one is apart from them?
Yes, it's possible. But the limb length theorem essentially says that we will take the limb length will be equal to the minimum value of this. Right?
But if
The
there
limb
are
length
like,
problem basically comes to compute the length of a limb in a simple tree by fitting of tree fitting an additive matrix. The input is an additive matrix D, an integer J, and the output is the length of the limb connecting the leaf J to its parent. All right. So here the limb is the distance between your organism, right? Or your leaf to the parent. Because you know the distance of the leaves with each other, right? You don't know the distances of the leaf with the parent. This is what we convert the problem into. Is this clear?
Yes, sir.
Yes, sir. Yes.
So now what you do is you apply this theorem and now you basically do the same thing. You calculate, right? So in this case, if you want to calculate the limb length of uh, chimpanzee, right? Chimp with its parent node, you will take all possible combinations and you see that the smallest distance is 1, right? It can appear twice as well. So the limb length of chimpanzee to its parent is 1, right? So you know that this value is 1. And as you have seen from the tree that we have fit, this value is actually 1, right? So now given this idea, we will solve this, we will try to solve, uh, we will try to fit a tree using this idea, using what's called additive phylogeny. And I will do this in next class. Briefly, what you do is you find out the shortest limb length, all right? And then you remove that limb and that node from the tree. All right, I'll quickly show you how. So let us say that this was your matrix. The minimum limb length for this, for J, was calculated to be 2, right? What you do is, by applying those formulas that I showed you earlier, solving the linear equations, what you do, because J is now absent from the matrix, you just remove J and the length of J from all the distances, right? So now the matrix that you would be essentially doing is, let's say we pick up arbitrary leaf J, the minimum limb length is 2 here. You remove, subtract 2 from all the other limb lengths, right? Because we don't know where this J, how the connection is happening, but this, we let us say that this J is not present anymore in our tree. We get what's called a bald tree, a tree which has basically an edge of length 0, right? And now you recursively do this, all right? And you keep on doing this until you get a tree with all the distances found out. All right. So this is the basic idea. This is the algorithm. In the next class, I'm going to go deeper into how this algorithm works. Before that, I stop the class. Are there any questions that I can take?
Yes, sir.
Yeah.
Sir, about the quiz, it is scheduled for next week.